Our next session for our In the Pipeline series is covering investigational products targeting mitochondria or the heart. But first, we're going to hear from Dr. Scavina on both topics.
In dystrophic muscle, there is an increase in what we call oxidative stress, or a strain on the muscle. This leads to an influx of calcium as a result of the disrupted membrane. The influx of calcium and other factors can negatively impact the mitochondria, better known as the powerhouse of the cell, because they produce the chemical energy cells need to function. This influx of calcium makes it more difficult for the cell to carry out its functions properly. As with other downstream targets in Duchenne and Becker, we can potentially improve the mitochondria through various approaches, such as enhancing the generation of mitochondria and reducing factors that negatively impact their function, such as the influx of calcium. If we improve the function of the mitochondria, this can help improve the overall function of the cell, even while the muscle membrane is still fragile. It is important to remember that the heart is a muscle too. The same disruptions to the muscle membrane and challenges we see in the skeletal muscle, such as an influx of calcium, necrosis, fibrosis, and disrupted mitochondria, all impact the heart muscle as well. These factors contribute to the dilated cardiomyopathy we see in Duchenne and Becker, where the heart becomes enlarged and weak. This can lead to heart failure. We can't focus on just the skeletal muscle, but must also look to see what compounds can be utilized to address the cardiac muscle. We must strive to understand how improvements in skeletal muscle impact the heart. By using compounds targeted to the heart to combat problems such as fibrosis or scarring, we may be able to fight the progression of dilated cardiomyopathy and improve an individual's quality of life.
Thanks, Meena. Joining us today on the panel is Ranzi Somarante from Imperium Bio, Gerard Marek from Stellis Pharma, and Larry Markham presenting for Cumberland Pharmaceuticals. As a reminder, if you have questions during the panel, please submit them to the comment box below your player. All right, we're going to start with Ranzi from Imperium Bio with the first presentation.
Thanks very much. Thank you very much. It's nice to present again this year. Thanks for having us back. Let's go to the next slide, please. So as we've said before, we are in the midst of an open-label phase 1 study in patients with Becker muscular dystrophy. The study has fully enrolled, and we're actually expecting our first subject to finish his 1 year on treatment coming up in the next couple of months. This is an open-label single-arm study. It's a dose escalation design where the patients are stepping up through 3 different doses while we assess them for safety and tolerability at each dose escalation. We're also doing biopsies of their leg muscles, looking at various biomarkers. We're doing cardiac MRIs at baseline 6 months and 12 months amongst a host of plasma biomarkers that will help us identify endpoints, doses, and other aspects of future trial designs. We're also conducting a number of physical function assessments, which, as you know, are very important in drug development in this field. Next slide, please. Our platform is based on mimicking a recently described sterile hormone called 11-beta-hydroxypregnenolone, or pindrolol. We believe that this hormone is released by mitochondria in response to chronic physiologic stress and is a way of helping the body adapt to this stress. Our drugs are mimics of this hormone, and in our human and preclinical experience, we can see that it modulates mitochondrial function coupled with tissue regeneration, and putting those two things together raises the point that was made in the video just a short while ago. And if we can make improvements on these two aspects, we believe that these may become valuable therapies for patients with Becker muscular dystrophy. Next slide, please. So these are older data. These are actually not from patients with Becker or Duchenne muscular dystrophy. These are patients with type 2 diabetes and ischemic heart failure. These are elderly patients from a VA hospital treated with an earlier form of the drug that we now have in the clinic. They were treated for 12 weeks and underwent biopsy of the quadriceps muscle before and after treatment. On the left side is an electron microscopic image of the before treatment muscle, and you can see that it's a disorganized muscle structure, and mitochondria are very difficult to make out. On the right side is representative of what the investigators saw after 3 months of therapy. The muscle structure is much more organized, and the mitochondria, are, while they're not completely normal in their appearance, are much larger. Two independent electron microscopists reviewed the images and came to the same conclusion that at least visually there was an improvement after treatment. Next slide, please. Along with the visual evidence, the investigators also measured key proteins in these patients. Again, these are type 2 diabetes patients with heart failure. These are Western blots from proteins obtained from the leg muscle. The B lanes on the Western blots are before and the A lanes are after. So if you see on the right side of the screen, key proteins such as dystrophin, utrophin increased after treatment with the compound. The other members of the dystrophin-associated protein complex also appeared to be more abundant after treatment. On the left side, it ties into some of the mechanistic evidence of this drug. You can see that key regulatory proteins such as myostatin and follistatin are moving in favorable directions, and we can see that proteins associated with differentiation of muscle stem cells and maturation of into more mature forms are also moving in a favorable direction. So mechanistically here, at least on the molecular level, the drug seems to be having some effect at various levels. Next slide, please. This is some updated information. While that study is going on in the clinic, we have done our first MDX mouse model with EPM01. Of course, this is a Duchenne-like model that's conducted in mice. We used what's called an in vivo eccentric injury model. As you remember, patients with Becker and Duchenne muscular dystrophy tend to injure their muscles when performing eccentric forms of stress on their musculature. This is an in vivo model, and these mice were treated for 2 weeks with EPM01, given by oral gavage twice daily. And we had 8 mice per dosing group. The way you assess these data is after the mice are treated for 2 weeks, they undergo an eccentric injury type of stimulus, and then you look for how much force is lost after invoking that eccentric injury stimulus. In the table, you can see that the wild type mice, these are the mice without a mutation, lost just a little bit of the muscle force. That's not surprising. The vehicle MDX is the untreated MDX mouse, and that mouse lost on average 65% of the force generating capacity of the leg of that particular muscle. And in the EPM01 arms, the force loss was somewhere in between the wild type and the untreated mouse, and the dose number 4 reached statistical significance. So this is encouraging to us in that the drug appears to be having some protective effect from eccentric injury in a model that doesn't require dystrophin. So we think this again ties back into the stress response aspects, particularly in restoring mitochondrial health and function. And I think next slide is my conclusion slide. So to summarize, our approach is attempting to couple bioenergetic improvement with tissue regeneration and potentially other effects. Our phase 1 dose escalation study has completed enrollment. The first subjects will be finishing their 1 year on therapy towards the end of the summer, early fall. Our early MDX mouse data suggests that there is some protective effect against eccentric injury in a dystrophin-independent model. We're going to be doing more work in this regard as we look further into deploying our platform into Duchenne muscular dystrophy. I think I have one more slide. What we do is not simply not possible without all the time and effort put in by our study volunteers and their families, and to the dedicated team of investigators and research staff who got this trial up and running and fully enrolled despite all of the restrictions from COVID. So I thank everyone.
Thank you, Ronzi. Really quick follow-up question. Well, first, PPMD is very proud to fund the early work in epicatechin and see this move forward. We talked yesterday a lot about the Becker population. We're grateful that you're studying in Becker. This is an underserved population. How are you thinking about the outcome measures of this population as you look towards the studies to show efficacy?
Yeah, as you know, Becker is a highly heterogeneous population, and in some patients, and there appears to be a threshold effect where their baseline dystrophin level may not have an impact on their clinical outcomes, and some patients progress faster than others. So as we think about future trials in the Becker population, we're looking very, very closely at the functional measures that we're testing. We're also looking at other functional measures and speaking to probably every opinion leader and thought leader and patient group that we can find in the next few months. So this is an important piece. I'd love to tell you that we have the problem solved in terms of how to contend with heterogeneity in an outcomes trial and an approval trial. But we're not there yet, but we're working very hard to reach a solution.
Thank you so much, and we appreciate that. And we plan to bring you all together, companies working in this space, so that we can all share learnings and move the field forward for that population. So our next speaker is Gerard from Astellas Pharma. I'm going to hand it over to you now.
I'd like to thank PPMD for asking us to tell you a little bit about our program. Next slide, please. Just the disclosures that I'm going to be talking about ASP0367, which was previously known as MA0211 when it was developed by Mitobridge, which Astellas acquired a few years ago. And ASP0367 is an investigational drug. It's not available for use outside these clinical studies. Safety and efficacy have yet to be established, and there's no guarantee that it'll receive regulatory approval down the road. Next slide, please. I'd just like to mention some key features of our current ongoing study. This compound is a PPAR delta modulator, and I'll say a little bit more about that in a second. But the ideal is that ASP0367 may act inside the muscle cells to increase the mitochondria's ability to produce energy. Because the mitochondria, as we heard about in the opening video, ends up being damaged and not fully functioning in your muscle cells. The phase 1 study in healthy adults has been completed, and we currently have a phase 1b study in boys with Duchenne muscular dystrophy that's open in the United States. It includes boys of 8 to young men of the age of 16. And the primary endpoint is actually safety and tolerability. The 6-minute cycling test is a new test that's sort of somewhat analogous to the 6-minute walk test. That's one of our secondary measures, in addition to the PUL and another other test as well. And we have received orphan drug designation from the FDA. And if the safety and preliminary efficacy signal in this current study is observed, then we'll certainly be investigating efficacy and safety down the road in phase 2 trials. Next slide, please. So as I mentioned, this drug may actually improve muscle energetics. And so we're studying this compound to determine whether it can really sort of turn on a switch in the cells that regulates mitochondrial function. That switch is called the PPAR delta pathway. And the PPAR delta receptor is something that our molecule actually activates, sort of like a key in a lock, more or less. And this PPAR delta pathway then regulates mitochondria by turning on different genes, generally, often genes that are related to energetics are producing energy in the cell. And when this pathway is on, the mitochondria use more fatty acids more often, and also more mitochondria are made. And using more fatty acids is important because you get more energy out of fatty acid breakdown than you do just breakdown of sugars. And so by using more fatty acids for energy, you have increased energy production, and the hope is that this will improve your muscle health and function. And because of this unique mechanism of action, combination use of ASP0367 with exon skipping therapies and or corticosteroids are possible, and we're doing that in this present ongoing study. Next slide, please. And this is just a schematic of the study. And there's first a 4-week screening period, up to 4-week screening. And then 2/3 of the subjects are randomized to being on active drug, ASP0367. And a third of the participants are randomized to placebo. Placebo being sort of essentially a sugar-like pill that doesn't have any active pharmaceutical ingredient. And so first there's a low dose period because the FDA asked us to look for a food effect in boys with Duchenne muscular dystrophy. And then we go to a higher dose period for a 10-week period for a total of 12 weeks then. And then at the end of this period, all subjects have the opportunity, if they wish, to go on for another 12 weeks on active ASP0367. And this is followed by a 4-week follow-up period. And about half these visits in this study are going to be ones that will be done in terms of home healthcare, where the nursing staff is specially trained to, in terms of not putting anybody at risk in this COVID era. Next slide, please. So who can participate? As I mentioned earlier, boys ages 8 through 16, having a diagnosis of DMD, taking stable dose of corticosteroids for at least 6 months, having some trouble walking quickly as determined by the 10-meter walk test, but you can move your arms up at least to the shoulder height. Exclusion criteria, having an illness, infection, or other condition that will not allow you to participate in the study, having problems with a heart not pumping well, electrical conduction problems, or having high levels of cardiac troponin I on a lab test, as well as having used certain types of drugs in the last 4 weeks, having kidney or liver problems, or currently receiving an investigational drug. And you can refer to clinicaltrials.gov, NCT 04 18 48 82 for more complete information. Next slide, please. So just to give you an update of where we are right now, so this is a phase 1 randomized double-blind, meaning neither your study doctor nor you will know which compound you're on for that first 12 weeks, whether it's active drug or placebo, and it's a placebo-controlled trial. The primary purpose is to evaluate the safety and tolerability of this drug. The study will also evaluate how your body processes the drug with blood levels, pharmacokinetics, and also how the drug affects function, namely using this relatively new arm cycling test. Trial sites are open now in the United States, and to find a participating site near you, you can visit clinicaltrials.gov and search NCT 04 18 48 82. In terms of the enrollment status, jumping forward from May to June, we've screened 4 subjects, and we've randomized 2 subjects into the study, and there have been no serious adverse events. Only 1 treatment emergent adverse event reported so far that didn't appear to be related to the study drug and resolved in 1 day. And I would like to thank everybody for listening to our talk, and now back to the studio.
Thanks, Gerard, so much. So just a brief follow-up question. We saw that endpoint of, or that outcome measure of the cycling test as a potential endpoint in the trial. This is something the community might not be as familiar with. Can you describe how you landed on that as the chosen measure and what it is exactly?
Yeah, we landed on it just because of sort of concern about being able to enroll younger boys, just given the interest in gene studies, certainly. But this arm cycling test is exactly that. Instead of using your legs to sort of turn the wheels at whatever rate you're able to, you're using your arms. So that's why it essentially is an arm cycling test. And essentially it's looking at then more so endurance rather than just simply peak strength, but looking for endurance. So obviously that sort of fatigue that you feel is one of the things that we want to hopefully tackle with our medication, and we're wanting to test certainly.
Wonderful. Thank you so much. Okay, with that, we're going to move on to Larry Markham for our final presentation, and then we'll get to the panel discussion. Larry?
Hey, thank you, Eric, Ryan. It's a pleasure to have the opportunity to speak before you on the FIGHT DMD trial. Next slide. I don't have any personal financial conflicts or disclosure related to Cumberland Pharmaceuticals. Next slide. So ifetroban, by activating the thromboxane prostanoid receptor, is profibrotic, meaning it induces scar tissue in the heart and lungs. And so the receptor, as shown in the cartoon, can be activated by several different ligands. And ifetroban is an oral once-daily medication that selectively blocks this receptor. Safety has been well established, but these with no serious adverse events reported in non-Duchenne patients and healthy volunteers. Next slide. So I began work with collaboration with FIGHT DMD Foundation, Cumberland Pharmaceuticals, and James West and Erica Carrier at Vanderbilt. And we investigated 3 MD muscular dystrophy mouse models and showed that survival in all 3 models was significantly impacted and improved by treatment with ifetroban. The graph on the top shows that cardiac function was stabilized. Next slide. There was a recent work that came out from Texas A&M University in early 2021, which showed that thromboxane receptor activation was enhanced or high in young muscular dystrophy mice, and that coronary vasoconstriction was measured in the presence and absence. And as the arrows show, ifetroban treatment is represented by the triangles, that ifetroban reduced vasoconstriction in young MDX in both aged and control mice. And this is important because one of the hypotheses, in addition to cardiac fibrosis, we also think there is an element of injury which occurs in muscular dystrophy, such that elevated troponin levels are one measure of heart muscle injury. And we feel that that has some contribution to the development of cardiomyopathy. So this would suggest that ifetroban could have a role in both fibrosis and also coronary vasoconstriction. Next slide. So working with Cumberland Pharmaceuticals, we submitted an FDA orphan product grant, and this is the first trial in Duchenne, not only in Duchenne, but focused on cardiac, which is funded by the FDA. The trial is also supported by the Marlin family and the FIGHT DMD Foundation. I will also suggest that Parent Project Muscular Dystrophy was vital in its consultative role in discussing the trial protocol. Next slide. So the study design is with the objective to evaluate efficacy, safety, and pharmacokinetics of ifetroban. The trial was designed with patients in mind to make it not too much different than routine clinical care for the 12-month duration. Treatment is daily oral ifetroban. 2/3 of patients will be randomized to drug and 1/3 will receive placebo. And we will stratify patients based on their cardiac function. So some with more moderate disease and some with mild disease. There will be an open label extension available to all participants. Next slide. So showing the key designs of the study, as we pointed out, there are 3 visits, one at baseline, one at 6 months, and one at 12 months. There will be cardiac measures, pulmonary measures, as well as some assessments of strength. Pharmacokinetics will be evaluated, but after the first dose, those can be performed at home. And several things that I would like to point out. All Duchenne boys older than 7, which really relates to their ability to undergo cardiac MRI, are eligible, regardless of mutation type, ambulatory status, or steroid use. All of those individuals would be eligible. There's no upper age limit, and standard of care medicines, including some gene therapies, will be allowed. There's no biopsy or 6-minute walk test. I think a lot of individuals have expressed interest in the study. However, there is some concern about waiting for other gene therapies or missing out on other trials. We designed this trial of a rather short duration, looking for a very strong signal of cardiac benefit. Next slide. So you can see that currently 8 sites are open and available for screening and enrollment. We have an additional 2 sites which will be opening very soon. And as an enrollment update, 3 of the 48 patients have already been enrolled and randomized. So we're ready for anyone who is interested. And here you see the sites which are available. Next slide. And so I'm available for questions or anything that comes up. Thank you again for this opportunity.
Great, thank you so much, Larry. So just a quick follow-up here. Do you see this being used as a monotherapy or in combination with standard of care heart medications at the end of the day?
Yeah, I think the steps from here in my mind are if there is a strong signal, then I think it needs a larger scale trial. But I would see this as part of the cardiac therapies that we already know. So ACE inhibition, aldosterone inhibition, and then you can put this on top of all of that to have a very aggressive, multifaceted approach to cardiomyopathy.
Great, thank you. And I noticed on the approved medications, you can add Amondys, Viltepso as well to that listing, right, of those who are able?
Yes, some of those were not approved at the time the trial was created, but those will be rolled in, and we discuss all of those therapies that could potentially be available.
Thank you.
Yeah, that's great to see. We're always happy to see those approved therapies included. All right, so yeah, we're going to start some questions. Again, if you have any questions, please submit them to the comment box below. So the first, Larry, you touched on already that there is an open label extension for that trial. So I'm going to ask you first about what dose is that on, and then I'm going to come around to both Ranzi and Gerard to see if there's open labels for those studies as well. So Larry, do you know if is it the high or the low dose that's offered in that?
Yeah, I think the open label extension dosing will be based on any signal that we are receiving from the 6-month visit. So that will be determined.
Okay, great, thank you. Ranzi?
Yeah, we, as I said, our first patients are starting to come off the first year of therapy, and we would very much like to continue collecting data on these volunteers. So we do hope to roll out an extension study for this trial in time.
Great, thank you. And Gerard?
Yeah, we have a 3-month open label period after the double-blind placebo-controlled part, yes.
Okay, great. And then this next question is towards Imperium. So since you've already fully recruited for this trial, the question is, what opportunities may there be in the future? Do you have a timeline for, when there would possibly be a phase 2?
For the Becker population?
Yes, for the.
So this is a 1-year trial. Our last few patients enrolled in January of this year. So I think we want to see the full year of data, and then we'll make a decision. But probably not until 2022 or slightly after, if we are able to go into a phase 2. Yeah.
Okay, great. Thank you. This one for you, Gerard. So has your product been studied in any other mitochondrial disorders? And if so, have you been able to take any lessons from that?
Well, we just recently have begun a study in primary mitochondrial myopathy. So we don't have any results of that. But one of the things I wanted to mention that in that study, one of the centers is actually looking at skeletal muscle and cardiac muscle imaging. And we do have some skeletal muscle imaging in our DMD study at about half the sites. So that is one of the things that we're hoping to learn from both of these studies, yes.
Great, thank you. And Ronzi, so you mentioned that you're collecting cardiac MRI data
yes.
in your trial.
Yeah.
So are you also collecting that on skeletal muscle and looking at things like fat fraction, as we talked about the other day with Dr. Vandeborn?
Yeah, no, not in this study, but we've been doing a lot of investigations and speaking to a lot of folks about MRI, muscle MRI. So in the next trial, I would very much like to use muscle MRI. Right now, we're going to focus on the heart because we're doing so many assessments on these patients, and it's a time commitment for the sites and the patients. We wanted to try and keep the assessments down to a minimum and try to get as much information from those.
Great, understandable, yeah. Patient burden is always a bit challenging in these clinical trial designs.
That's right.
And Gerard, are you all considering, so right now you're looking at this 8 to 16 range within this
Yes.
first study of about 18 patients. Is there going to be an opportunity, in the future where you maybe look at some of those younger ambulatory patients? Obviously, as you discussed, there is a challenge. We have a lot of trials in that space, but, if that would be something you're looking at.
I think that when we move into phase 2, that's what we probably would be doing, where we sort of might be running essentially sort of a basket study where the patients that are younger, where we're looking at lower limb function, would go into that study, and the older ones where there's somewhat limited in terms of lower limb function would be exploring upper limb function. Yes.
Okay, great. Thank you.
Larry, I got a question for you. Given the drug you're studying, what about the Becker population? Given the cardiac issues within the Becker population, are you looking at them as a potential?
So there have been a couple of areas where we've discussed as sort of obtaining a signal of benefit, and then Becker would be ideal just as well. But then also, I think, carrier mothers. So that is a population that, having a largely cardiac phenotype, this could be something that would apply for them as well.
Great, and actually Gerard as well. Are you thinking about Becker?
Well, at this time we aren't, but certainly down the road, that is one of the things that would be sort of the next thing that we indication that we would go into would be Becker's. Yes.
Okay.
And definitely happy to hear that that consideration of carrier mothers. I mean, as we've funded some studies at Nationwide Children's, that is another underserved population. So always good to hear that they're being considered. Larry. Another question is kind of, you've talked about looking for a strong signal here in this first study. if you look towards a pivotal trial, and this may be a hard question to answer, have you started to think about, or have there been conversations with regulators about what kind of, an outcome measure could be used? This has been a pretty big challenge in Duchenne, and it's something that we're constantly exploring. We're working on, as part of our guidance as well, kind of these issues. So just would love to kind of hear your thoughts and see, what you're thinking.
So we, another FDA-funded grant. So we've had conversations with the FDA, and my colleague, John Soslow at Vanderbilt, got an R01 funded by the FDA. So in 1 year, we figured out that we had 2 FDA-funded grants in Duchenne focusing on cardiac. So what we hope to do with his study, which is really in effect a very detailed natural history study of some of these detailed MRI cardiac measures. And so we're partnering with Nationwide, Children's National, here at Riley and Vanderbilt to enroll patients to look at what is the natural history and what are some meaningful cardiac endpoints. And so we hope to use that data to then drive that discussion with the FDA of, okay, we have clinical trials, but now we actually have hard and fast and true data that says these endpoints are meaningful, and this is what we would like to put forward as an endpoint for cardiac trials. Interestingly enough, we are using the same MRI protocol in both, so that if there is something that we determine in the natural history study, we will have that data being obtained as part of the clinical trial.
Well, great. We love to see that cross collaboration. Hopefully there's some really good results that come out of that.
Yeah, and as we're developing the guidance as well with cardiac, I think this is all really, really important. We're adding cardiac to the update to the Duchenne guidance, so such important topics for consideration as we move forward.
And then, Ronzi, so you kind of, I think, alluded to this a little bit when you asked that question of whether I was asking about Becker or Duchenne. So are there plans with EPM01 to look towards a Duchenne population? And if so, I mean, again, the question is always, timeline.
Yeah. Well, as I just showed you, we just did our first MDX mouse study with EPM01, and for us, that's our first finite step into that. We're going to do another study. I think we'd like to learn a little bit more about the drug characteristics and dosing in that population. There are a couple other animal models I want to conduct, but we're trying to move in that direction. We just need to be very thoughtful and methodical about it, because what we do take into a clinical trial, we need to feel very good that we're doing something meaningful.
Yeah,
great.
Absolutely.
Thank you.
Larry, I'm sorry to come back to you on this ACE-ARB question. You had mentioned, if approved, these would be used in combination, but what about for the trial? Are patients allowed to be on an ACE?
Yes.
Okay.
Yes, they will stay on their existing therapies through the trial. So they just have to be on it before they enter the trial.
Okay. Thank you so much.
Great. Gerard, I wanted to kind of touch on one part that we noticed in the trial design, where you noted that about 50% of the visits at home are at home. Does that include functional testing? And was that as you were preparing to launch your trial, during the pandemic? Was that a response to that, or was this kind of always in the trial design and something that you're thinking about as you look at future trials and trying to kind of minimize that burden to patients?
Yeah, when COVID hit hard, early last year, we actually stopped many of our trials, especially ones that hadn't started yet. So we were sort of redesigned things, and so it was redesigned in that manner. So that unfortunately we were only able to limit the actual clinic visits to about 50% because of certain things that we actually, in terms of looking at motor function, it wasn't really realistic to do that on a home basis. So that was the limitation in what we were able to, and moving towards that. Yes.
Okay, great. Thank you. Yeah, we've been having more and more discussions about kind of what that is going to look like, what lessons we're going to learn from COVID. And I think finding ways to make these trials easier for patients to participate in, ones that maybe get away from the trial sites so we can have more inclusion. It is always a challenge, especially as you're working with the older patients and non-ambulatory. Those travel distances can be pretty tough on that patient population. So we also appreciate that kind of consideration in trial design. Larry, you had mentioned that, and it was up on your presentation as well, that as part of the study, it was from an FDA orphan product grant. And this is something that I think maybe the community is not as familiar with, and it's one of the first ones we've had in Duchenne. Could you maybe go a little bit more into what that means and kind of what those interactions look like?
Right. So, the FDA, very similar to the NIH, has what's called an RFA, which is request for applications. And those come out and they may be very disease specific or condition specific or technology specific. And so this just happened to come out at the time in 2017 when we were exploring how to fund a study such as this. And so Cumberland Pharmaceuticals had researched this mechanism or funding mechanism. And so we decided to apply that route. And fortunately, we're successful. So I think in my mind, it has been very fruitful so far in that there is review and back and forth with the FDA on the progress of the trial and how it's going. This doesn't necessarily guarantee any pathway to approval or anything like that. It's just this is a funding mechanism to allow the trial to be paid for.
Great, thank you. And Ronzi, Ryan had mentioned some of those earlier studies with epicatechin carried out by Craig McDonald and some others. Were there any takeaways, or either from biomarker data that was shared, or any other kind of lessons that you have been able to kind of take and implement, as you've thought about these designs and just your learnings in this population? Oh
yeah, no, absolutely. In the work that we've done with Craig and was supported by PPMD, we've identified follistatin as a potential biomarker for efficacy of the drug. As well as myostatin. We've looked at nitrates and nitrites because the drug appears to be upregulating nitric oxide synthase activity, which of course in the peripheral musculature would be a good thing because these patients have relative ischemia in their peripheral muscles. So on the biomarker side, we've learned quite a bit, and we're using the same biomarkers in the current study. So yeah, that work has been foundational for where we are today.
Yeah, just to follow up to that, I know we've talked about, so how was that trial defined for Becker? Functionally, was it by mutation, was it age? This is always
the current study
question. Yes, sorry.
Yeah. So we did a lot of work with Craig McDonald in identifying this population. So from a mutation perspective, they do have to have a Becker or Becker-like mutation. And so functionally, there are, and you can look at our listing on clinicaltrials.gov, there are cutoffs for the 6-minute walk test and the 10-meter run walk. So we're trying to recruit sort of a mild to moderate Becker population. And the mutation has to be verifiable. So those are, I would say, the two most character defining or population defining criteria that we have.
Great. Yeah, and you're paving the way for other trials, so I just can't say enough. We're grateful for that, and we love to see these underserved populations. And we also love to see the companies looking, and investigators and academics looking at the heart. We know the heart is a muscle too, and as we extend function, we're always asking the question, is the heart built to last? So.
Yeah, and then Gerard, we had one last question. I think someone noted that it said there was some accelerometer testing within your study. Could you just maybe tell us a little bit more about that in the trial design?
So we're working with Actigraph, and the whole point of that is just to sort of monitor continuously over in sort of real-world manner motor function over a day. So that's essentially what we're doing. So it's a way of actually bringing in measures actually at home and not simply dependent upon coming into the site for a visit and then being tested over that period in which one's worried about how one's going to perform. Yes.
Oh, great. Thank you. And we'd like to thank all of our panelists for joining us today for some really good discussions. We really appreciate it. We're going to move on to our next Pat Chat here. Joining us today, we're going to have Pat joined by Dr. Beth McNally, who's the Ward Professor and Director of the Center for Genetic Medicine at Northwestern University. So they're going to be discussing the heart, which, as we've talked about, is something that we're going to make sure that is included in our upcoming guidance. So we're going to have a new section on cardiac to tackle some of those really tough issues that we've kind of been talking about. So with that, I'm going to hand it over to you, Pat.
Hi, Beth, and welcome. So good to have you here.
Hey, Pat. Great to see you.
Thank you. So, Beth, you've been working on dilated cardiomyopathy and Duchenne and Becker for a very long time. And I think that every parent worries about the heart, right? Because it's a muscle, and obviously in the cardiac MRI, we hear news such as you see strain early as 5 years old. Well, what does it mean? Can you help shed some light about what we know about the heart and the heart disease in Duchenne?
Yeah, I mean, you're right. The heart is a muscle, and it is affected in Duchenne muscular dystrophy. But different from skeletal muscle, at least the way I approach it is we have so many more medicines to treat the heart than what we have for skeletal muscle. Now, we're excited. There are many things coming in the skeletal muscle domain, and we have some drugs approved now. But you know, the heart and the type of problem that happens in the Duchenne heart is actually a problem we know very well in cardiology. And we actually have quite a lot of medicines and devices and approaches to treat that problem. And so I think the key part, as any parent is thinking about this, is to just be very proactive, make sure you have a cardiologist who is familiar with Duchenne and the management, the skeletal muscle side of management, as well as the cardiac side. And that's just really important to have somebody knowledgeable about and keeping up to date about what those treatments are.
Do you recommend early cardiac MRIs?
Well, MRIs are always a little bit tricky in the very young ones, because if you require sedation to sit still for an MRI, there's risks associated with sedation. And so luckily, most of the time where we see some of these early changes, it's around the age when most children don't need sedation to do the MRI. So I think that's encouraging. I think many people still remain pretty reluctant to be sedating kids for cardiac MRIs really early in life. And so, you know, part of it has to do with your child and how readily they can sit still and do an MRI. And MRIs can be a little bit scary. You're in a big machine and it makes a lot of noise. So.
Yeah, absolutely. I think that, you know, MRI is suggested to be the gold standard. And I think what gets really confusing is a parent has an MRI and then an echo and tries to make sense of those two tests to say, are they apples and apples? And what can you interpret from each of those? Could you shed a little light on both of those?
Yeah, really, really good question. So echoes, I will say both echoes and MRIs are both very much user dependent. So again, you want to be in a facility that's very used to doing these. And for example, echo is highly dependent on the echo technologist who's holding the probe and taking the images. And they can do even very young children and do echoes if the tech is patient enough and there's something there for the child to be distracted by, or sometimes even a little baby, they can do them on with the parents holding them. So echoes are very, very safe to do, but to do a really quality, good echo on especially a small child, it takes time. And so you just want to make sure nobody's rushing through that process and they're just taking their time and getting the images they knew. Now, you had mentioned strain. And so again, a really good echo, we can see strain differences in really good echo. That's not always done because we do that probably more on the adult side than on the pediatric side, but a good echo lab actually can see some of those really even early changes by echo. But the bonus of an echo is that it's safe, it's really easy to do, and it can be done even on very, very young children. MRI, as you mentioned, gives us more information. It gives us more of the tissue quality, whether we see what we call delayed enhancement or some of that scarring that's present on the heart. And again, probably can see strain a little bit easier, although some of my echo colleagues will argue with me about that and say we can see it just as well on the echo side in the same. So they're complementary tests. I do notice on the pediatric side, they will order these pretty regularly, once a year or so, even when a child's growing. Lots of things can change. As we get to the adult side, we probably don't do them quite as often. One, because it can be really hard for an older Duchenne patient to do an echo. To do an MRI because of just the need to be flat in the scanner. Whereas some of my older Duchenne patients, we can do great echoes in their chairs and not even have to move them. So that's a good thing. So a lot of it has to do on what your facility, what their experience is, how good are the images. I still get lots of great echo images, and we do that all the time.
Good. Beth, I know you're an adult cardiologist, and so this isn't meant to be, just in your view, as a mom with a child, would you, and I'd come to you with a 5-year-old and say, I'm going to start cardiac meds. Would you agree with that or not?
I think they're pretty benign to be on. If it were me, I would probably say yes, particularly around the ACE inhibitors or angiotensin receptor blockers. I think we know they're extremely well tolerated. I work very closely with our pediatric cardiologists. I actually go and do a family clinic with them. So we work, there are many children who get treated with these medications, and it's a real mainstay for any children with kind of children onset cardiomyopathy. They do this all the time. Very safe, very well tolerated. We usually start with one of those, and then we start talking about adding a beta blocker at a little later state. And that's where I think the pediatric cardiologists don't embrace beta blockers as readily as we do on the adult side, where we pretty much believe everyone should be on a beta blocker because it's good for hearts, manages blood pressure beautifully well. But again, the good news is these are medications that we have decades and decades of safety data. We know very well that they're well tolerated and very safe. And what's kind of cool on the adult side is we've also seen several new medications actually come into our management now. We are now using newer drugs. Probably people have seen some of them being advertised on TV, but this sacubitril valsartan is a newer drug known as Entresto. That works very well for heart failure patients, really remarkable results. And then our other new drug that we use now quite readily in the adult setting is medications that are used for diabetes, which are called the Flozin type medications, which have turned out to have remarkable efficacy for people with heart failure and reduced ejection fraction. So we're starting to even think about using those as well.
And Beth, you're doing a loop recorder study. Can you talk about the loop recorders and should we all have one?
Yes.
And do you think there are arrhythmias in Duchenne? All of those in one?
So loop recorders are, what we do know is that there is generally, when we look across hearts and we see more scar on the heart, we know that that's an increased risk for having irregular heart rhythms. And irregular heart rhythms can affect the top chamber of the heart in the form of atrial fibrillation, as well as the bottom chambers of the heart in potentially more lethal rhythms that we call ventricular arrhythmias. And so we don't want those to happen. We want to treat them if that's a possibility. We want to prevent them. And so our best defense against those is monitoring for it. And so many people are now more familiar with the patch monitors, which you can wear for up to 14 days. Those are outside the body, and it's a skin patch. It sits on you. Those are now being used in the pediatric setting. They weren't for a while, but now they are more readily being used there. And 14 days is pretty good. Like I get watching somebody for 14 days and knowing that they haven't had anything in 14 days is great information. And then when we do see a little bit of something, or we're not quite sure and we want to get more information, but we don't want a child to have to keep wearing a patch 14 days after 14 days after 14 days, we instead have these little capsules that can go under the skin and allow us more ready monitoring pretty much 24/7. And you can do regular downloads of those little loop recorders. Though what we hope to do is correlate imaging data from MRIs with the data that we're getting, not just off loop recorders. We're actually doing it sometimes on people wearing patch monitors, because we can do those more readily. And all our patients now have the patch monitors, so we can increase our sample size a little bit that way. And we'll be asking that question. So we know Duchenne hearts have a lot of scar on them, kind of more than what we're used to seeing, but we don't necessarily think of the arrhythmia risk as being much worse than other cardiomyopathies. So that's really the central question of the study, which is to try to make those correlations. What we see on imaging, what does it correlate with the rhythms, so that we can better guide our patients about whether they need devices or not.
Yeah, I think one of the questions with gene therapy is, should the gene therapy improve skeletal muscle function? Is the cardiac target useful, and does it increase any risk to the heart?
It would be one of those things I would call a gaping hole in the literature right now. It is something. It is in many ways experiments that are being done real time. And, as we've all lived through pandemic this last 16 months, we've all been living through what it is to be in experiments real time. But for a parent who has a child with Duchenne, that's a bad situation to be in. And we need to get more information. We can get a lot of information from our animal studies where we can model this, and we're trying to learn more about it. Is gene therapy going to equally treat the heart and the skeletal muscle, or is there going to be disproportion there? We have some concerns about some of the animal studies for antisense treatments, and those again are in use right now, but we know they don't quite get into the heart cells as well, and what's that really going to mean long term? And so I think anybody who's been receiving those treatments, if you've been in a clinical trial or if you're actually on an approved drug, I think really getting together and gathering that information so we can get as much gathered and studied and make recommendations as quickly as we can. I think that's what we want to be doing. We really have to learn more about this.
Thank you so much, Beth. I think the Action Network is collecting this data. Hopefully we will get together, collect the data, and really understand this for kids. We're going to come back to you, and so thrilled that we're going to be working on the guidance with a specific heart cutout. We have so much to do there. Thank you so much for joining us tonight. Appreciate all you do.
All right, take care.
Speak soon.